Hey guys, welcome to Chapter 17, Topic 1. In this topic, we're going to focus on intermediate filaments. Chapter 17 is all about the cytoskeleton. So we have 4 different topics in this chapter. And in the last topic, we're going to wrap up with muscle contraction. So we kind of bring together all the different topics we talk about in 1 through 3. And as I said, in Topic 1, we're going to focus on the intermediate filaments. So our outline is, what are intermediate filaments? What do they do, and what's their role in the cytoskeleton? It's a very brief topic. As always, here's our objectives. If you have any questions about these, please let me know so we can go over them some more in class. But before we dive into what are intermediate filaments, let's talk about what is the cytoskeleton. So the cytoskeleton basically is, we think of it like the bones of the body, right? But that implies that they're rigid and they don't move. So they're really more of a combination of the muscle and the bones of the cell. They're really flexible, they allow for movement, but they do provide structure. So it's really important that you understand that aspect of it. They're not stiff like our bones in our body are. And they can take on a variety of shape, and their main role is to help organize interior components, and they have some other aspects too where they interact with the environment. They carry out coordinated movement. If the cell is capable of movement, it's the cytoskeleton that helps do that. And as I said, it's a dynamic structure. So once again, remember that the cells, that having this cytoskeleton is what allows our cells to move and be flexible. Otherwise, we would have significant problems if anything happened that required our cells to stretch or bend. So let's get into what are intermediate filaments. This is the first of the filaments we're going to talk about. The next 2 are in their own separate topics. So intermediate filaments, they're rope-like structures, and you can see a picture of that on this slide here. And as you can see, they have this central alpha helical region followed by 2 heads on either side. And these are bigger than actin and they're smaller than myosin, so they're kind of the intermediate, which is why they're called intermediate filaments. And you can see on the slide it says they're 10 nanometers in diameter. I'm not super worried about you knowing how thick each of these filaments are that we're going to talk about, but I do want you to understand that. The central amino acid sequence is standard within intermediate filaments, so it's always going to be the same. And what you can see here is they form these rope-like structures. So we start with a monomer, and then we get into a coiled coil, and then we go into a tetramer with coiled coil. It's basically like how we make rope. It's very, very much like how we make rope, except for with the big key being that we have these heads and the head and the tail regions that you can see here vary a lot. And so that variation is what allows intermediates to have variable functions. So remember, it's very much rope-like. It's a lot of coiled coils on top of each other, those alpha helices. So what kind of microfilaments are there? We talked about how there was variation from the head and the tail region, but the middle is pretty clear. And you can see here that there's 4 main types of microfilaments with two groupings. There's the cytoplasmic one, so keratins, vimentin, and neurofilaments. And these all have very different functions, and we're going to go through each of these here in a second. And then we also have intermediate filaments that are localized specifically to the nucleus. And these are in animal cells, and they make the nuclear lamina, and we'll talk about that one here in just a sec as well. So here's where we're going to look at each of these groups. So let's start with keratin. And I'm sure you've heard of keratin before because it's in most of our epithelial cells, and it's something that we extract regularly, especially if you're not doing a good job of being clean in the lab. This is something that can come up in protein extractions frequently if you have contaminated it with your own cells. So keratin's a very diverse class, and it's in, as I just said, epithelial cells, and it spans the interior of the cell. And what it does is it helps that cell stretch, and you can see a picture of that here on the slide. It helps it maintain that elasticity. Otherwise, we would have the cells breaking apart from each other as things stretch, which makes sense as to why keratin would be in our skin cells. Vimentin's another one, and that one's we find in connective tissue, and it helps anchor organelles, and once again provides that flexibility. So if organelles are anchored, they're not going to move around too much, but because the vimentin is still flexible, it allows for a little bit of stretching and movement within the cells. In neurofilaments, we see these intermediate filaments, these neurofilaments, serving as a way to strengthen the axon of the nerves so that they aren't damaged as easily. And then lastly, we have our nucleolaminins, which are in the nuclear membrane, and they help strengthen the nuclear membrane. And this reinforces that to make sure that the nuclear membrane is nice and strong and it doesn't break apart very easily. So what are these intermediate filaments doing in the cytoskeleton as a whole? And as you can tell, based on our last slide, a big thing is that they're reinforcement. They help make sure that the cell keeps its shape, but allows for flexibility at the same time. And it also is, it's another important aspect of it is that it anchors organelles. And then there's this last part. We didn't talk about this too much in this topic, and I don't want you to focus on it because it's not a huge thing. But they also participate in cell-cell junctions through the pores known as desmosomes. And we haven't really talked about that this semester, so I'm not super worried about that. The big take-home I want you to get from intermediate filaments is that they serve as reinforcement, but also allow for flexibility within the cell. So this is the end of a really short topic on intermediate filaments. As I said, as always, review the topic objectives and let me know if you have any questions about intermediate filaments. And when you're ready, move on to topic number 2.